Genetics is the fascinating science that explores how traits and characteristics are passed from parents to their offspring. This field delves into the similarities and differences we observe among family members and even across entire species. By studying genetics, scientists uncover the laws that govern heredity, helping us understand why we inherit certain features, such as eye color or height, and why siblings can look so different. Genetics is not just about appearance, it also explains the transmission of diseases and other inherited conditions. Through this study, we gain insight into the very blueprint of life, setting the stage for deeper exploration into how our bodies function and evolve. Gregor Mendel, an Austrian monk, is celebrated as the father of genetics. Born in 1822, Mendel's early education took place in a monastery where he developed a keen interest in science and mathematics. Despite his aspirations to become a teacher, Mendel faced setbacks, failing his teaching certificate exam. However, his curiosity led him to groundbreaking experiments with garden peas, where he meticulously observed how traits were inherited from one generation to the next. Mendel's work laid the foundation for the laws of inheritance, which remain central to genetics today. His perseverance and scientific rigor transformed our understanding of heredity. Genetics is not just a theoretical science, it has powerful applications in our modern world. One major application is genetic engineering, where scientists alter the genetic makeup of organisms by introducing new genes. This technology has revolutionized medicine, agriculture, and industry, enabling the creation of disease-resistant crops and innovative therapies. Another vital application is genetic counseling, which helps families understand and manage inherited diseases like hemophilia, thalassemia, and sickle cell anemia. By matching the genetic profiles of prospective parents, some genetic diseases can be prevented or minimized, improving health outcomes for future generations. Heredity is the process by which genetically based characteristics are transmitted from parents to their children. This concept is at the heart of genetics, explaining why offspring resemble their parents and how certain traits are preserved or altered through generations. Heredity not only covers physical features but also includes the genetic constitution of an individual, which influences everything from metabolism to susceptibility to certain diseases. By studying heredity, scientists can trace the lineage of traits and predict how they might appear in future generations, offering valuable insights for both medicine and evolutionary biology. While humans share many common features as a species, there are countless small differences among individuals. These differences, known as variations, are what make each person unique. Variations can be observed within families, between races, or among different populations. They arise from the complex interplay of genes inherited from both parents. Some variations are subtle, like differences in eye color or height, while others can be more pronounced. Understanding these variations is crucial for studying evolution, disease susceptibility, and the diversity of life on Earth. In genetics, any inheritable feature of an organism is called a character, while the alternative forms of that character are known as traits. For example, eye color is a character and its possible traits could be brown or blue. This distinction helps scientists describe and analyze the diversity seen within a species. By studying characters and their traits, geneticists can track how specific features are passed down and how new variations arise, deepening our understanding of inheritance and evolution. Chromosomes are the structures within cells that carry genetic information from one generation to the next. Through advanced microscopy, scientists can photograph dividing cell nuclei and arrange chromosomes by size and shape in a chart called a karyotype. Each species has a constant chromosome number. For humans, it's 46. These chromosomes are organized in pairs, with each pair containing one chromosome from each parent. This pairing ensures the continuity of genetic information and is fundamental to the process of inheritance. In every organism, chromosomes occur in pairs known as homologous chromosomes. Each pair consists of chromosomes that are similar in size and shape, with one inherited from the mother and the other from the father. These pairs are crucial for sexual reproduction, ensuring that offspring receive a balanced set of genetic information. Homologous chromosomes carry the same types of genes, though the specific versions or alleles may differ. This arrangement is key to the diversity and stability of genetic traits across generations. Chromosomes are divided into two main categories: autosomes and sex chromosomes. Humans have 22 pairs of autosomes which determine general body features like height and complexion. The 23rd pair consists of sex chromosomes, designated as X and Y. Females have two X chromosomes, XX, while males have one X and one Y, XY. These sex chromosomes are responsible for determining the biological sex of an individual and play a crucial role in the inheritance of certain traits and disorders. The sex of a child is determined by the combination of sex chromosomes inherited from the parents. While the mother's egg always provides an X chromosome, the father's sperm can carry either an X or a Y chromosome. If an X-bearing sperm fertilizes the egg, the child will be female XX. If a Y-bearing sperm fertilizes the egg, the child will be male XY. This simple yet elegant mechanism explains the roughly equal ratio of males to females in human populations. Chromosomes are more than just structures. They are the carriers of genes, the true units of heredity. Genes are specific sequences of DNA that determine everything from physical appearance to bodily functions and even behavior. The term gene was first coined by Wilhelm Johannsen in 1909 to describe the hereditary units passed from parents to offspring. Each chromosome contains thousands of genes, and it is the unique combination of these genes that makes every individual distinct. Genes often exist in alternative forms called alleles. Each allele occupies the same position or locus on homologous chromosomes but can affect the same characteristic in different ways. For example, the gene for height in pea plants has two alleles, one for tallness and one for dwarfness. The dominant allele expresses itself even if only one copy is present, while the recessive allele is only expressed when both copies are recessive. This interplay between alleles is fundamental to the diversity of traits seen in living organisms. Every organism has a unique set of genes known as its genotype. The observable characteristics that result from this genetic makeup are called the phenotype. For example, in tongue rolling, the genotype could be RR homozygous dominant, RR heterozygous, or RR homozygous recessive. Both RR and RR individuals can roll their tongues, while RR individuals cannot. This distinction between genotype and phenotype helps scientists understand how traits are inherited and expressed. When discussing genotypes, it's important to distinguish between homozygous and heterozygous conditions. Homozygous individuals have two identical alleles for a trait, like RR or RR, while heterozygous individuals have two different alleles RR. This difference affects how traits are expressed and inherited. For instance, a homozygous dominant individual will always show the dominant trait, while a heterozygous individual may carry a recessive allele without expressing it. Understanding these terms is key to predicting inheritance patterns. Tongue rolling is a classic example used to illustrate inheritance. By examining family pedigrees, scientists can track how the ability to roll the tongue is passed from parents to children. This trait is controlled by a pair of genes, and its inheritance can be predicted using genetic charts. Such examples make the principles of genetics tangible and relatable, showing how simple genetic rules can explain everyday differences among people. The Punnett square is a simple yet powerful tool used to predict the possible genetic combinations in offspring. By placing the gametes of one parent along one side and those of the other parent along the other, the square shows all possible genotypes that can result from their union. This method helps scientists and students visualize how traits are inherited and calculate the probability of different outcomes, making genetics more accessible and understandable. Some traits are inherited through genes located on the sex chromosomes, a phenomenon known as sex-linked inheritance. Disorders like hemophilia and colorblindness are more common in males because the genes responsible are found on the X chromosome. Since males have only one X chromosome, a single recessive gene can cause the disorder, while females need two copies. Understanding sex-linked inheritance is crucial for diagnosing and managing these genetic conditions. Gregor Mendel's experiments with garden peas were revolutionary. He chose peas because they had many varieties, bred true for generations, and could be easily cross-pollinated. By carefully controlling which plants bred with each other, Mendel observed how traits like flower color and seed shape were inherited. His meticulous records and analysis led to the discovery of the fundamental laws of inheritance, which form the basis of modern genetics. Mendel summarized his findings into three fundamental laws. The law of dominance states that one trait can mask the presence of another. The law of segregation explains that gene pairs separate during gamete formation, ensuring offspring inherit one gene from each parent. The law of independent assortment reveals that genes for different traits are distributed independently. These laws have stood the test of time and are still taught as the foundation of genetic science. Mendel's principles have far-reaching applications. By understanding how traits are inherited, breeders can predict the outcomes of hybrid crosses and select for desirable characteristics in plants and animals. This knowledge is invaluable for agriculture, allowing the development of crops with improved yields or resistance to disease. Similarly, animal breeders use these principles to enhance livestock traits, contributing to food security and economic growth. Mutation refers to a sudden change in one or more genes or in the structure or number of chromosomes. These changes can have significant effects, sometimes causing diseases like sickle cell anemia, where a gene mutation leads to abnormal red blood cells. Mutations can also be triggered by external factors, such as radioactive radiation, which was tragically observed after the atomic bombings in Japan. While some mutations are harmful, others can drive evolution and lead to new traits in populations.